All right, everybody, we're going to get started here at 7:15, so we stay on schedule. My name's Dan Cook. I'm a clinical instructor at the Vanderbilt University Medical Center, and my other chair over here is Elizabeth Kramer, who is an assistant professor of pediatrics at Cincinnati Children's Hospital Medical Center. We are going to talk today about non-pulmonary manifestations and various models to better understand the effects of CFTR in organ systems that don't include the lungs, but also to get a better context of how modulator therapies might play a role in these non-pulmonary manifestations. These are our session description and objectives, and with this we have a great panel of speakers today. As you can see from the titles, we vary in terms of what the origin of the model is going to be and the organ system that we're looking at. To start off, we're going to have Dr. Doug Bartle speak. But before he comes up and I give him his introduction, I want to remind the speakers of ground rules that we have here. You have about 15 minutes here that'll start. You'll have a timer up here that'll keep track of that. A yellow light will go off when you have about 3 minutes remaining, and then it'll turn red when time's up. And we ask that you stick to that so that we can remain on schedule. Note for the speakers, your very first slide advancement needs to come from the computer, just technical reasons, and then you can subsequently use the clicker after that. We're obviously around to help if you need anything. So with that, I'm going to introduce Dr. Doug Bartles, who's the Executive Director of the National Ferret Resource and Research Center at the University of Iowa, who's going to tell us about the role of diet and pregnancy-associated morbidity in CF ferrets.
All right. Here. So good morning. Welcome to Phoenix and NACFC for the intrepid few that show up this early in the morning. Oops, I'm still on yours. My bad. There we go. All right. Sorry about that. There we go. So today I'm going to tell you a short story about diet, CF ferrets, and pregnancy. I do mention ivacaptor in the talk. This is a study not sponsored, but we do work with Vertex. And to start a non-pulmonary talk, I'm going to show a picture of a lung. So basically the short version is John Englehart and others have shown our CF ferrets have multi-organ CF disease, lung, pancreas, GI tract, liver, and so on, but that is not the topic for today's talk. The topic today is about the CF baby boom. This is lifted directly from the CFF registry and a very nice review article written very recently. We're topping out at about 600 pregnancies a year in CF. And as you can imagine, spread across many different centers, that individual cases in each center is actually going to be quite low. So to understand rare events in this population, we're going to need to do multi-center trials, which are obviously getting fired up. But there is one place where we have a lot of pregnancies all at the same spot, and that is in our ferret colony. So we have a very nice century of over the last few years, we've had well over 100 CF mothers give birth to kits and thousands of carrier ferrets giving birth to CF ferrets. So again, it's a nice way to try to get a handle on fertility and obstetrics in a population where the same people are looking at all of them. And we've already learned quite a bit. So we've published this before, but clinical doses of ivacaptor in ferrets cross the placental barrier and prevent meconium, which is a big plus. We can even dose the kits through the breast milk of the mother. It also gets through the breast milk, which is a big plus and helps us rear these. And even on the fertility front, we've learned a lot. So if you have CF activity in utero, the vas deferens is produced properly, and the males are actually fertile for the rest of their life, even after removal of ivacaptor or CF activity. We actually use pancreatic insufficient males as our major breeders at this point, because we've shown over and over and over again that once they get through in utero, they're actually fertile the rest of their life. And this is a similar story for females. The loss of CF activity does not affect them significantly. However, Again, if we're looking for rare cases, we did end up with an interesting observation. So we had 4 nearly identical cases over a 2-month period a few years ago. Again, these are CF ferrets. They're pancreatic insufficient. They've had about a 1-month holiday, or more than a 1-month holiday from ivacaptor, so their pancreas is gone, their GI tract is not functioning properly. All of your CF disease phenotypes are kicking in. And the second trimester, everything was going great. They're back on ivacaptor. They're on soft food with pancreatic enzyme supplementation. They're bright, alert. We palpated. They had large litters, more than 10 for each of these, which is actually above average litter sizes. And it was going fine until the first few days of the 3rd trimester. Suddenly, that next morning, they were lethargic, in appetite. They just were not doing well, sudden health decline. We looked at their blood panel. The liver enzymes were very bad. ALT was off the charts, and obviously we had to euthanize them at that point. I know it's a little early in the morning for this, but in that upper left-hand panel, you see what was obvious. The liver is orange. It should not be. That is major steatosis in these animals. And that's shown in the second panel where, again, this is an H&E stain of a liver. All that white is not supposed to be there. That is all just fat inside those hepatocytes. So these things are literally nothing but fat. So they have major steatosis. The other panel shows some other CF liver disease things, which we'll talk about at a different time. So trying to translate this back to the clinic a little bit. So there is a disease in humans called acute fatty liver in pregnancy, AFLP. In the normal population, it's pretty rare. It's only about 1 in 20,000 pregnancies. But intriguingly, it does increase the rate significantly as you have multiple births. So if you have twins, the rate goes up quite a bit. If you get triplets and above, which of course there are not that many cases of, the rate actually goes up significantly. So very similar to our large litter phenotype that we saw. And this is because late in pregnancy, for fetal-placental growth, you need to liberate a lot of fatty acids. So if you aren't getting nutritionally, you have to liberate your stores in the body, and this can overwhelm the liver. And as the hepatocytes in the liver are kind of the last vestige of a place to take it up, if you get too many in the bloodstream that aren't being used, they end up there, and you end up with this fatty liver disease. And this has also been seen in ferrets long ago, like 30 years ago this was reported. They called it pregnancy toxemia at the time. Intriguingly, it was also correlated with large litters and hepatic lipidosis, or fatty liver again. But again, 30 years ago, they didn't even have the standardized chow for ferrets, so their diets were very wonky back then. And the reason why I keep saying diet is in carnivores, examples, pets of cats, dogs, ferrets, if they have a malabsorption problem or even anorexia for a few days, they get hepatic lipidosis, which again, as you see, all that white is just fat in your liver. So again, if they no longer get enough calories, they start liberating the fatty acids from their stores. It overwhelms them, their livers get destroyed. So even within a few days of not eating, you can get major lipid or major liver problems in carnivore pets. So the obvious hypothesis here is our CF ferrets have a fatty acid and malabsorption issue. They're pancreatic insufficient. This isn't a huge stretch. And this is exacerbated by the increased need in late pregnancy for extra fatty acids with large litters. So this gets us to, this gets us to, can we fix this? So obviously drug interventions on all this is one way to try to do it, but the other is, can we just fix this with diet? And what we ended up zooming in on is lysolecithin or lysophosphatidylcholine. Because it's already prehydrolyzed, it makes my cells that get absorbed in the GI tract even in the absence of pancreatic enzymes. So the hope is we can basically increase free fatty acids by adding LPC to their diets. Obviously, it's a common emulsifier in food and additives in both humans and in pets. So we have 50 years of safety data on this, that it's fine to feed this to people and animals. And when we look at sources of LPC, sunflower seed, there's plant-based ones, which are normally what humans eat, sunflowers and others. We obviously, as a carnivore, we wanted to stick to that. So we ended up going with dried egg yolk, which is about 40% LPC. And as you can imagine, you can buy metric tons of this stuff from a bakery for not very much money. So it was a great additive to our diet. And what was kind of intriguing as a side note, obviously, as we start looking up diets, CF, egg yolk, you end up running across from the 90s and 2000s and earlier, all the CF moms talking about how to get their kids to gain weight in the pre-HEMT era, and egg yolk and hollandaise sauce popped up all over the place. So we thought we rediscovered what everyone already knew. So we're basically making hollandaise sauce for ferrets. And as this was the fun for my team. So I gave them an ingredient challenge. You have 3 ingredients: ferret chow, a non-lactose milk replacer, and dried egg yolk. Now make this where your very picky judges will like it. So we literally made different combinations of this, gave each of the ferrets a 3-bowl challenge, and wrote down on a notebook, every one of them, like which ones they liked. And different amounts of water was the main variant, where we went from kind of chunky chili all the way to melted ice cream. And surprisingly, I was betting on chunky chili. Melted ice cream won. So is what it was. And basically our single serving dose of this EMF diet recipe is shown below. And obviously the first one, we just wanted to test this diet. What is a single serving for this diet? Even though our fat ratio wasn't that much different from what we started with, we weren't sure how to dose this. So what we did was a pilot study. Each of these lines is an individual pregnant ferret over time. And we measured the amount of food left in the bowl to see how much they ate, and basically did a quartile analysis. So as you can see, each of those individual lines in the second trimester, you can see they only eat about 25 to 50% of that single serving. As they get a little into the third trimester, they're eating 75% of it. And once they're nursing, they basically clean the bowl, so that was good. One version of this, we changed the amount they eat each day, which is actually a little complicated for our rearing crew. But then what made us feel much better is once we either transferred the kits to a new foster mom or they weaned, the mom quit eating. She went back to, not quit eating, but went back to normal 50% levels. So they basically are self-regulating their intake, which makes feeding them 1,000 times easier than having to actually measure out each day what everybody needs. And this has borne out over the past year. We've now expanded this diet to our whole colony, and our rate of obesity in the whole colony has remained unchanged. So they're self-regulating the diet, which makes simplicity of feeding much easier. And obviously, simplicity and ease is not what we're looking for. What we're looking for are positive results. And what we got were very positive results. So shown here in blue is the survival rate of kits from 1 week to 10 weeks of life. Historically, with what we're calling the historical diet, which was kibble with cat food, dog food mixed in. And as you can see, the survival rate was in the 50 to 60% range, which historically we thought was actually quite good. However, when we switched to EMF diet, losses between week 1 and week 10 completely disappeared. So if the mom is taking this EMF diet, she's nursing these kits, we literally do not lose any of them. So over a 3-month period, we lost 1 out of very many. EMF through the mother's milk is great. And we also had a similar effect when we looked at them post-weaning. Now they're eating the EMF diet themselves. Historically, as everyone in this room can imagine, CF ferrets were slightly smaller than their siblings. Weight gain was a little slower, overall size was a little lower. That all disappeared when we switched over the diet. With the EMF diet, there is literally no difference between a CF and a non-CF ferret anymore. They have the exact same growth rate for both males and females. Similarly, when we take away ivacaptor in these ferrets, they have again, as you could imagine, their GI tract is no longer working as well. They're now pancreatic insufficient. Even though we supplement them with pancreatic enzymes, they still kind of get a tummy ache and kind of quit eating for a few weeks, and they basically stall out in their growth rate historically. And that's shown on the left-hand panel, where you go for multiple weeks where they don't gain any weight. And again, in the EMF diet shown on the right, it's now percent weight increase instead of overall weight. But EMF diet across males and females was just spectacular. They didn't even slow down. They just kept on growing. So both when nursing, when weaned, and after removing ivacaptor, the EMF diet basically made the CF overall weight gain and size defects basically disappear. So obviously that was not what we set out to do with the study, but we are spectacularly happy that with a simple change in diet, we've actually simplified our process significantly. But the question was, back to where we started, does this cure our acute fatty liver in pregnancy? So obviously we had quit breeding pancreatic insufficient ferrets when we saw those original results. So we took about a year hiatus, figured out all the stuff with the diet. Once it was up and working, we basically restarted and repeated exactly the same experiments. 1 month ivacaftor holiday or more, pancreatic insufficient moms, bred them, crossed our fingers, and monitored them thoroughly to make sure we weren't having any issues. Short version is 0% mortality, no clinical signs of disease. We monitored them actually multiple times a week for ALT levels, no change, couldn't even see a blip. We were assuming we were going to see some kind of subclinical, couldn't even see that. It worked great. And then, as we mentioned earlier, that litter size seemed to be a major issue, where if you got above 8 or 10 before, it was basically a death sentence. All of these had more than 12. One of them had 16 kits, which is like extraordinary. And not only did she successfully rear them without a problem, all those kits survived as well. So it is working spectacularly. So small change in diet, huge effect on outcome. Obviously, this gets us to what the overall effects were. Again, small change in diet prevented this pregnancy-associated mortality. It normalized weight gain across multiple stress points in the CF life while nursing, post-weaning, and with removal of ivacaftor. And I don't have time here today, but it's on the poster. We did look at overall healthcare utilization by these ferrets, and historically, as you can imagine, CF ferrets needed a lot of vet care and a lot of extra work. It was 70 to 80% of every animal sometime within a month needed something. Now that rate is basically zero. As you can tell, they're growing fine, they're happy, everything. So our total amount of time we need to spend to rear these has actually dropped significantly just with the change in diet. This gets us to the final part of what this means for the clinic. As I've mentioned at the beginning, obviously in the lung and the pancreas and others, the ferret has modeled human disease quite well. Whether it models obstetrics, I have no idea. Again, the GI tract is very different in the ferret. Obviously, 12 kits in a litter is very different than a human, but it was worth kind of spreading this and giving everyone else information on this, just in case you do see some weird case that, oh, I remember this, the ferret guy told me this might be a problem, kind of thing. And the only thing I'll say is, obviously, I'm preaching to the choir here, but you should definitely have nutritionist support for all CF moms. That's all I'm going to say. It actually can have a huge effect. And I look forward to discussions with all of you as we get more and more case studies put together in the human field, and I'll be looking forward to seeing, hopefully, no interesting results and all good things on that front as well. And with that, I just want to acknowledge everyone in the broader Englehart lab and everyone who works on ferrets, and spectacular job implementing this and getting it to work great. And with that, thanks.
Thank you. That's fantastic. Can everyone hear me? Yeah. Okay, I guess I'm just shy.
Yep.
We do have a microphone in the room. If there are any questions, you can also input questions into the app, and then we can all see it, and we can even vote questions up and down. If you go to our session in the lower right corner, there'll be a Q&A button you can press, so please feel free to go to the microphone. I'll start us off with a question.
Yep.
Were you able to look at lipids in the blood or other signs of metabolism?
We looked at it a little bit. We weren't quite set up. And the first time, obviously, we weren't expecting it, so we didn't have all the data historically. And then when we redid it over again, we normalized it so much that we took all the samples. We never even ran them because it obviously
You did a
worked.
good job.
It worked. It worked at a certain point. And since we didn't have the comparator, it was hard to know what abnormal looked like because we were no longer doing
Absolutely,
it. So.
absolutely. Well, it certainly will influence me to recommend more hollandaise sauce in my
Exactly.
CF clinic, which I hadn't thought of. Do you think that the withdrawal from ivacaftor had any role in it? I mean, obviously in terms of the modulator effects of it, but also that the fact that they were on it and then withdrew from it, we've certainly seen some, in patients, some poor outcomes upon abrupt withdrawal. Do you think that played a role? Or
Yeah, so we have a lot of experience with that, obviously.
Yeah.
It's pretty standard at this rate, and we've actually presented it at prior meetings, basically the timing of when in life you remove the ivacaftor has a huge effect on outcomes. Obviously, very early in life, they just don't thrive because GI tract. Well, maybe with EMF, of course, the problem is that's all historical data before this diet, so it's a little hard. But then there is a pancreatic stress point that if you take them off too early, they have pancreatic problems. Later, they have another weird metabolism. You do it pre-wean. What we've come around to is, as you can imagine, is shortly, a couple of weeks after weaning, when they've stabilized from the weaning, that's when we pull them off. And it seems to in a broad population, not really. What is normal now is basically no major change at that point. But so there might be, but it's now kind of buried in such large numbers we don't see it anymore. So.
We've got a question in the chat asking, have you looked at changes in mother's milk composition as a result of the diet?
That we looked at drug levels in the milk, but we've not looked at the actual changes in the mother's milk composition.
And I have a question to follow up on that.
Yeah.
How does ivacaftor levels in either the serum or the breast milk change with the diet?
It increased it because, again, it's a high-fat diet.
Yeah.
So yeah, it's not surprising. And again, this gets us to, if we can combine both the dosing and the food together, is what we're obviously trying to get to. So.
Well, thank you.
All right.
Quick question.
Oh, one more question. Sorry.
David Stoltz, Iowa. Thanks, Doug. It was really nice. And it's good to see it's easier to do than before.
Exactly.
Two questions. One, did the liver histology improve when you changed the diet? And second, with these animals were sort of healthier, did that impact any of the other disease phenotypes
So
in the
both,
animal?
very good question. So on the liver side, we did have a few of them that successfully reared that then we ended up taking down a few months later, looking to see if there was any subclinical steatosis. If you squinted really hard, maybe, but then again, we don't have enough numbers of what normal looks like. We think it's probably in the normal range of CF-related liver disease in those animals. As per, what was the second one was, just overall health. So again, the worry was, now that we no longer have a growth defect in the ferrets, do we actually screw up liver disease progression and pancreas liver progression? And so far, so good. It looks like they're healthier, and we have a bunch of ferrets that have chronic lung disease with Pseudomonas, but they look perfectly happy while doing it. So that's, I think we solved one without causing a new problem of less disease. But again, that will bear out as we get larger and larger studies if we start seeing a little bit of a decline, but we haven't seen it to date. So, all right. Thank you.
Fantastic. Next, we have Dr. Lynn Hajar from the Cleveland Clinic in Case Western Reserve, who's director of the Notobiotics facility there, and she'll be speaking on gut microbiota and CF. I'll get you set up here. There you go. Thank you.
Okay. Try and get a pointer here. Okay. All right, thanks for the opportunity to discuss our results on gut microbiota contributions to intestinal disease and CF. I have nothing to disclose. And I guess about a decade ago now, it was shown that there is a high relative abundance of E. coli in the feces of children with CF. And here, each thin line is an individual, and the thick line is the average of all those children. Another way that this was shown here is, again, this increase in proteobacteria in children with CF, and E. coli is a proteobacterium compared to control feces. And also there was this decrease in the bacteroidetes, so there's less light blue in these samples. And so, and it also was shown that higher E. coli was associated with higher fecal calprotectin, and that's secreted from neutrophils, so an indication of inflammation. And in more recent studies, this higher E. coli was also associated with a shorter stature in infants or in children. And so, suggesting that E. coli might be pathogenic. And other evidence of intestinal inflammation in people with CF, again, includes this increased fecal calprotectin, although that's also associated or higher with pancreatic insufficiency, so perhaps malabsorption. Capsule endoscopy has revealed inflamed regions in the intestines. And, maybe 30 years ago now, there were these differential sugar absorption tests that showed gut barrier leakiness, and of course, dysmotility. And more recently, with longer survival, there's an increased incidence of colorectal cancer that could be due to inflammation, although the mechanism is unknown at this time. And question I've always had is, which came first? Did the dysbiosis or the high E. coli lead to inflammation, or was there inflammation there that then allowed the E. coli to grow? And it's really hard to study in humans, and so chose to use germ-free mice, because there are all the confounding factors in humans, like antibiotic use, diet, and genetics. And so I guess some of my lines disappeared, but in studies several years ago now, so we made the germ-free CFTR knockout line, the S489X mouse, and then we transferred wild-type microbiota into the mice and either control or CF mice, and then followed them for 1 to 3 months and looked at the microbiota, and we found that the CF mice selected for a community that mimicked CF with decreased overall alpha diversity and higher E. coli. And then further, when we actually took CF microbiota and gave it to new germ-free control in CF mice, we found again it's maintained in the CF mice, but surprisingly it reverted to a non-CF-like state in the control mice. And so this kind of made it harder to study because you couldn't just put CF microbiota into a wild-type mouse to see what happened. And so the actual data I'm going to show you from this paper, basically we did this first set 4 separate times, and we combined the data, spread them over a year, and then we used cecal content from this last experiment in this transfer down there, and we call these the X and Y samples. And basically, this is the data that showed that it was the genotype that was dominant over the microbiota. Just in terms of alpha diversity, the inoculum itself obviously was the most diverse, had the greatest number of different taxa, whereas the CF recipients had the lowest diversity. And when we look at how these branch based on a dissimilarity Bray-Curtis analysis, you can see all the CF samples from CF mice branch together, and all the non-CF ones branch with non-CF mice. And this sample 4 here was the inoculum that was CF inoculum used in these X and Y samples, again showing that it's diverged away in the control mice. Other studies at the time, we were looking to see could we measure intestinal inflammation. And because the small intestine has a defect where the Paneth cell granules get entrapped in the thick mucus, so I had our veterinary pathologist look at Sika to try and make sure she was blinded. And really, there was no difference in just gross histological H&Es, staining, looking at all these factors between CF and control mice, whether they were conventional mice or germ-free. And if you actually look at the individual components and the different mice, nothing was remarkable that jumped out. But one component that we consistently saw was an increase in Th17, systemic Th17 cells. So if you gauge on take the whole spleen, look at the CD3 positive cells, and then gauge on the CD4 cells, and stimulate with PMA and ionomycin to measure the interferon gamma versus IL-17 production, we found that there were higher numbers of these Th17 cells in the spleen of the CF mice, whereas there were equivalent numbers of Th1 or gamma producers. And similarly, in the mesenteric lymph node that drained the intestines, we saw these higher Th17, but equivalent Th1 cells. And here, even in unstimulated cells, they're already making IL-17 directly ex vivo. But unfortunately, our mice had SFB, the donors of those microbiota. So when I moved to the Cleveland Clinic, basically I decided to use a more defined community to study this further. And so I chose to use the altered Chaeter flora, which is a small community of just 8 bacteria that's been used for a long time in mice. These are all mouse isolates, mostly firmicutes, but there's 1 Bacteroidetes in this community. And when we look in the spleen and lymph node, while the spleen was not significantly increased, we did see higher Th17 even with just this very small community in the mesenteric lymph nodes. And if you look at this in context in terms of numbers of cells, though, there weren't very many of those cells. And this is, something maybe you're not supposed to do, is combining flow data from 2 different institutions spread over many years. And so this is really kind of very preliminary, because if you look at the group numbers, some only have 3 mice per group, but basically this is we have germ-free mice, mice monocolonized with E. coli, mice that have the whole altered cellular flora, 8-member community, or altered cellular flora with E. coli. Then the FMT mice, which are the ones I just showed you in our publication, those are the fecal microbiota transplant study, or the conventional mice that we were breeding at the University of Washington here. So maybe it's an increase in these cells with increasing complexity. But when we had ASF plus E. coli, we found actually we had a lot of these Th17 cells in the mesenteric lymph nodes of our CF mice. Whereas in the control mice, we did not see that. Surprisingly, when we only had monocolonized E. coli, the control mice now had significantly higher, even with just these 3 mice, Th17 cells in the mesenteric lymph node, but not in the spleen. And I'll get back to this observation of high MLN Th17 cells in a few slides. So we looked at a measure of gut inflammation is that fecal calprotectin in the mice. And when we looked in our ASF colonized mice, we did not see higher fecal calprotectin in the CF mice. And then we gave them E. coli, and a week later looked at the fecal calprotectin, and still that was not higher, although these values are pretty low. Excuse me. So the question was, though, how are we getting these systemic Th17 cells, and could it be due to barrier leakiness? So we thought, we actually set up this experiment to look at intestinal permeability. So at the end here, we did the fitsidextran assay. And basically, this is giving oral fitsidextran, which is a large molecule that will usually stay in the intestines. But if there's more leakiness, you'll be able to detect more in the serum when you bleed the mice 4 hours later. And so Craig Hodges shared his data with us that he had gone with SPF mice, showing this higher serum fitsidextran in the SPF CF mice. And, surprisingly to us, we found that the ASF plus E. coli colonized mice had a similar level of leakiness, if you will. But then when we looked at germ-free or mice colonized with only ASF, we found that those did not show increased barrier leakiness. And furthermore, then we looked at our bacterial load in these mice. And so we counted E. coli, CFU, and MacConkey agar. The ASF members cannot grow on the MacConkey agar aerobically. And so at necropsy, we just took content from the large intestine, and we found that there was, significantly higher counts of E. coli in the colon of the CF mice. And similarly, or surprising, I guess I wasn't sure if we would see anything in the mesenteric lymph nodes, but we saw just decent number of E. coli, CFU in the CF mice, but also the control mice had a few counts. Now, when we coming back to what I talked about, those monocolonized E. coli mice, we found that when we monocolonized mice with E. coli, the control mice now had high counts of E. coli, and whereas when they have ASF there already, the counts are much lower. But they now are equivalent to what we see in our CF mice. And so as a reminder, these are the mice that had small amounts of Th17 cells in the mesenteric lymph node, although not systemically. And then we looked at the ASF community by 16S ribosome RNA sequencing. And we've initially noticed that if we just looked at the amount of DNA, so this is nanograms of DNA per milligram of feces, our samples from ASF colonized mice had just a lot less DNA and than our SPF mice. And this is likely, because the community is, it's not as dense. There's only so much that these 8 bacteria can grow to. So, but when we look then at the composition of this community, again, we see this higher E. coli in after the mice have been colonized with E. coli. So here it's like a 30% relative abundance. But even before we give them the E. coli, we saw this significant enrichment of this, well, what used to be one of the lactobacillus, now it's ligulactobacillus, or I'm not sure how you pronounce that, but which is, again, also found in human CF, the higher lactobacillus. And we also saw that the Parabacteroides, which is one of the Bacteroidetes, was depleted in the CF mice. So these are features of ASF that are consistent with what's seen in human CF samples. So in conclusion, I guess I went faster than I thought, but despite identical and defined initial colonization, E. coli is definitely more abundant in the CF mice colonized with ASF, but ASF alone does not appear to increase intestinal permeability, and you really need that ASF plus E. coli to now see higher intestinal barrier permeability and these systemic Th17 cells. And again, this was a commensal E. coli organism, and so potentially suggesting that it can be somewhat pathogenic in the CF context. We also showed that the non-CF mice are able to limit E. coli to the mesenteric lymph node without the systemic Th17 cells. And then in terms of my question of which came first, the dysbiosis or inflammation, I think the data suggests that the dysbiosis even precedes inflammation, at least in terms of that ASF community where you already see a shift in the microbiota before there's even any change in permeability. And so future directions, I'm obviously interested in determining what's driving this dysbiosis at the beginning. And Craig Hodges has a couple of these great models, mouse models, where CFTR, there's a floxed allele of CFTR, and then you can express Cre with the villain Cre, using the villain Cre promoter to express, to delete. Actually, you can have the either delete or turn on CFTR in intestinal epithelial cells. And we've made the off mouse germ-free, and we're in the process of beginning these studies there to see if that's sufficient to drive the dysbiosis. And as far as permeability, we're also interested in determining what is it that drives, like in E. coli, what could be driving the increased permeability when E. coli is there? What about host factors? Is it the inflammatory response now to this E. coli, or that's driving this increased leakiness? And then, everything we've looked at is in the content of or feces. So what about at the mucosa? What's happening there and in the lamina propria itself? And then another part is, can we add back Bacteroides that we saw the Parabacteroides depleted in CF? Would that help reverse this E. coli expansion? And perhaps more distant future directions is looking at the role of microbiota and obstructions in CF. And so just want to thank my colleagues at the Cleveland Clinic and at Case Western. And of course, all the work that I showed, the unpublished stuff, was performed by a former technician, Alex, and undergrad student, Lauren. They were really amazing. And of course, I want to thank the Notobiotics crew that maintains all these germ-free and notobiotic mice. And I need to thank the mice and, of course, funding from the CF Foundation. Okay, thank you.
So as a reminder, you can input questions into the app or ask them up at the microphone. I'll go ahead and start. When looking at the E. coli that's recovered from the GI tracts, is there any differences in how, for say, the metabolism of that E. coli, or how they would typically respond to the environmental cues that they're growing in?
Good question. I haven't looked, and I have no idea. But obviously, so one thing that we are in the process of doing is we'd like to do some of this kind of a dual RNA sequencing So taking a section, I mean, I'm particularly interested in the distal small intestine where there seems to be a lot happening there, and looking at the host, what's the host RNA response and what's happening in the bacteria as well. And I think with these defined communities that are sequenced, it's much more tractable to look at that.
Long time no see, Doug. So you talked about probiotics, obviously. Have you looked at prebiotics? Could you actually, is it too much fat not getting absorbed? Is it other small metabolites? Can you actually overwhelm it and fix it?
Yeah, that's exactly. I don't know. I mean, that's partly, by knowing what's driving the dysbiosis, that might help. So is it the weird mucus that's made that's providing a different nutritional input, or exactly? I don't know, but that would be another, I mean, we would definitely want to explore that once we understand how it's arising.
I have another question. So I would have naively thought that antibiotics were driving a lot of the dysbiosis in our CF patients. Have you incorporated that at all into this model system? Does it worsen the dysbiosis?
Yeah, that's a very good question. I too would think that, and yet even though you look at some of the human results, I mean, generally when they people report them, they try to keep them off of it, have the samples that they've looked at that they've been off of antibiotics for a month. But even with that, I would think that there would be some residual effects, and yet they pull out some of these changes that we're seeing. So I haven't done anything with antibiotics here. Actually, that's one thing I'm proposing to do, is to maybe, what if we treat and get rid of E. coli? Yeah. Can we reverse some of that leakiness or other things? So haven't done those yet, but yeah. Right. Yeah. Yeah, I really didn't pursue that at all. Besides just PCRing and showing it was there, and then dropping it and going for the way that didn't have it there. So I can't comment on it at all.
All right, excellent.
Thank you.
Thank you.
Okay.
So our next speaker is going to be Alice Eastman, who's a PhD candidate at the Department of Human Genetics at Johns Hopkins School of Medicine, where she studies single-cell transcriptomics using human sweat glands. She'll be telling us about the establishment of a primary sweat gland culture suitable for CFTR functional and RNA analysis.
Thank you very much. Hello, everyone. My name is Alice Eastman. I'm a sixth-year graduate student in the Human Genetics and Genomics Department at Johns Hopkins School of Medicine. I work with Gary Cutting and Neeraj Sharma. I'd like to thank the organizers of this year's conference and the moderators of the workshop for giving me the opportunity to talk today. I'd like to share the establishment of primary sweat gland cultures that are suitable for CFTR and RNA analyses. Okay. I have no relationships to disclose, and now it works. First, I'd like to introduce you to the sweat gland in its native environment for some context. Let me get the laser pointer. There we go. The eccrine sweat gland, shown here in orange, can be thought of in two parts. First is a secretory coil, which sits in the dermis or hypodermis of the skin. In the coil, sweat components, primarily sodium chloride and water, are secreted into the gland, making a primary sweat with a similar salt level to the blood. The other part is the reabsorptive duct, which leads through the dermis and epidermis onto the surface of the skin. And this portion is responsible for reabsorbing ions and nutrients, creating a final sweat that is hypotonic relative to plasma. To zoom in a little further, eccrine sweat glands contain 5 major cell types. Within the secretory coil, there are two layers of cells. On the interior are the dark cells and clear cells that are together referred to as secretory cells. And surrounding the secretory cells are the myoepithelial cells, which are spindle-shaped contractile cells that function to squeeze the sweat upwards. And in the reabsorptive ducts, there are also typically two layers of cells that we refer to as luminal cells on the interior and basal on the exterior. In people with CF, chloride is not reabsorbed into the sweat gland in the duct, which greatly increases the concentration of chloride in final sweat. Chloride concentrations of greater than 60 millimoles per liter are considered diagnostic for CF. Because sweat chloride concentration is an easy and non-invasive measurement to take, and this concentration will change based on CFTR function, it can also be used as a biomarker for modulator efficacy over time. And it generally correlates well to lung function. Our goal is to learn more about how CFTR and other ion channels localize in the sweat gland epithelium, and whether we can use sweat gland cells to test CFTR function and response to treatments for people with CF. The cell culturing technique we have employed for sweat glands is based on human nasal epithelial or H&E cell culture, which is a well-established technique that has been widely used for CF and other research. For this technique, we acquire nasal airway cells by brushing, suspend them in media, and then conditional reprogramming is achieved by plating the nasal cells on a feeder layer of irradiated 3T3 mouse fibroblasts and treating with ROCK inhibitor. Cells are expanded, then differentiated at air-liquid interface and on filters. And then these filters can go on to be used for functional measurements in short circuit current measurement, which is done in Ussing chambers. And I'll go into that in a little bit more detail soon. So to culture sweat glands using a similar conditional reprogramming tactic, we start with skin, which we are lucky enough to get from Johns Hopkins Hospital in the departments of dermatology as biopsies and the Department of Plastic and Reconstructive Surgery as incidentally removed tissue or surgical skin I have here. So we microdissect sweat glands from the skin and enzymatically dissociate them into a single cell suspension. We plate on the 3T3s and treat with ROCK inhibitor. They're expanded. Here we use liquid-liquid interface on the filters, and then similarly we can use the Ussing chambers for functional measurement. Here I just want to show you how we're getting these sweat glands and how we know that they're truly eccrine sweat glands. On the left is an image of two particularly distinct sweat glands. The skin has been dyed with neutral red, which makes them easily distinguishable from the surrounding dermis. I also want to point out that in this instance, we can visualize and actually dissect apart the ducts, which is not typically feasible. And next, I want to show you two images from another major part of my work, which is 3D immunofluorescence imaging of the glands and two-photon microscopy. So here I'm demonstrating that sweat glands we harvest consistently stain for eccrine sweat gland specific markers. Here, alpha SMA, stained in red in the middle, is a reliable marker for the myoepithelial cells, so we can distinguish duct from coil. And on the right, also stained in red, is keratin 10, so, which is a ductal marker. And we can see this also distinguishes the two parts. And you can even see in this one, sort of as the red fades along here, the transitional region between the two. So once we get the sweat glands, if instead of imaging them, we dissociate them into single cell suspension, we can then plate them as I described. So about a week after we plate the cells on the mouse fibroblasts and treat with ROCK inhibitor, we see small clusters of epithelial cells. Over time, these islands grow until eventually they push aside the feeder cells and reach confluency. Next, we transfer the cells to filters for differentiation. Here on the left, I want to demonstrate the final resulting eccrine sweat gland culture, which demonstrates a cobblestone morphology that has been previously described for similar cultures. On the right, for comparison, are our differentiated nasal epithelial cells on filter. So an important question regarding these cultures is, what is the cell composition? Since we're starting with dissociated sweat glands, which I mentioned earlier have about 5 major cell types, and we're taking them from the dermis, which has other resident cell types, it's important to know what we're looking at. Since another part of my overall project involved single-cell RNA sequencing of the whole microdissected and dissociated sweat glands, we were lucky enough to be able to use this technique on the cells grown on filter as well and compare them to the whole dissected glands. So here I'm showing you 2 UMaps from this analysis. In a plot like this, each dot represents a cell, and the distance between the dots corresponds to the similarity of the sequenced transcriptomes in the respective cells. You'll hear a lot more about this in the next talk. So we have similar cells that are clustered together, and by looking at what kinds of genes are upregulated in those separate clusters, we're able to differentiate distinct cell types. On the left, I'm showing the cell type distribution from the whole sweat glands, while on the right are the cultured cells from the filters. So here I'm demonstrating that we find both secretory cells and ductal cells in our cultures. You can also see that we have a high proportion of myoepithelial and fibroblast cells represented. But what we found is that these cells tend not to adhere well to the filters and tend to detach over time and come off with the media change. With the cultured cells, we want to see if we can detect CFTR function in these cultures. So, brief refresher on short circuit current measurement. Let's see. To briefly go over this concept, we take filters with the differentiated cells and mount them in Ussing chambers, which are equipped with electrodes to measure ion flux across the chambers on either side. Here we're starting with the same chloride concentration on the 2 sides, a symmetric buffer. When we add an activator of CFTR, like the combination of forskolin and IBMX, we see an increase in current as chloride crosses through CFTR. Then when we add a CFTR-specific inhibitor, such as INH172, we see a drop in current, which we can then quantitatively measure CFTR function. Here I want to show that our ESG primary cells derived from individuals without CF demonstrate functional ENaC and CFTR activity. So if you bring your attention to the tracing on the left, you can see that we're looking at current on the y-axis measured in microamps per centimeter squared over time on the x-axis. Carbachol addition, which you won't use for H&Es, activates the ENaC, which is then inhibited by amiloride. Then forskolin and IBMX activate CFTR, which is then inactivated by INH172. I also want to show you here that on the right, we're also getting activation of CFTR by a combination of cyclic AMP and ATP, which has been previously published in whole sweat gland electrophysiology. So both of these activators are functional in our system. Here I want to show that inhibition of CFTR can be achieved by either INH172 or GlyH101. And this is important because these 2 CFTR-specific inhibitors bind to different parts of CFTR. So this is supporting that this current is really CFTR-specific. So the traces I showed were from cultures derived from individuals without CF. But with this technique in hand, we recruited 2 individuals with CF, both of whom were homozygous for F508del, who were kind enough to donate both skin biopsy and nasal brushing cultures for us to, cells for us to culture. So first, here's what we saw in the H&Es. As expected, the blue line here shows that with no treatment, there's minimal activation or inhibition of CFTR. However, when we incubate with Alexa-Teza, Alexa-Caftor and Teza-Caftor, and then acutely with Ivacaftor, we see significant activation of CFTR, and then INH172 specific inhibition. Here I'm showing that this is a statistically significant increase, though we're not brought up all the way to wild type function. We get to about 60% of wild type function with the H&Es, and this is about exactly what we'd expect. And then for the sweat gland cells, here, these are from the same person. We can see that cyclic AMP increases current in both groups, the treated and untreated, with alexiteza. And we're attributing that to non-specific channel opening, mostly. We see a small increase in current with ivacaftor addition in the untreated and a more significant boost in the treated. And then the difference in current when we inhibit with INH 172 is more significant in the EDI treated. And likewise, I want to show that forskolin IBMX, here they activate the treated but not untreated cultures. And here we're using inhibitors, both INH 172 and GlyH 101, that both work here as well. So to finish up, we've developed a conditionally reprogrammed primary cell culture for the eccrine sweat gland. Sweat gland cultures demonstrate both ENaC and CFTR activity. ESG and H&E primary cultures derived from the same individuals with CF show similar function between each other and expected function. And I'd like to thank my lab very much, especially Erin and Audrey, my fellow grad students, who are very, very supportive.
Thank you. As a reminder, you can come to the microphone or put in your questions on the app. And I'll start out. I'm used to thinking of airway and nasal cells on these inserts, so this may be a naive question, but do these ESGs, are they secreting a liquid layer or that you can sample? Or
Right.
Yeah.
So there's not mucus
Right.
or presumably sweat.
Right.
No, there's no noticeable secretion.
Okay. And I assume if you do histology, the CFTR is polarized since you have such a beautiful listening chamber response.
Yeah,
Have you
yeah,
looked
they
at
do
okay.
polarize, yes.
Excellent. Excellent. Okay.
And to follow up on that, how much duct versus the other? Because they're going in opposite directions. I was trying to figure out.
Right, right. And that's complicated. So yeah, the proportions, and they're not stable, the proportions. So one thing that we're really trying to do is we also have cultures going that are the microdissected ducts. And I showed those ducts with a very distinct different portions, but usually it looks more like you have the big coil and then a little string. And you just don't get enough cells from the ducts alone, often, to make the culture. So we need just a few that have those distinct ducts to get those cultures going.
Hi, Alice. Oh, sorry about that. Hi, Alice. Can I ask which media you use to expand and differentiate these sweat gland cultures?
I am supposed to obfuscate that a little bit
Okay.
until we are published, which should hopefully be very soon.
Copy that. Thanks.
So the ESGs, obviously, you've gone to great lengths to make these an excellent model. How do you envision that these will be used as surrogates for other aspects of the CF
Right.
disease?
Right. So there are a couple ways. So first, there are a couple genotypes for which you see discordant phenotypes between, say, lung and sweat gland. So there are individuals who will have good lung function and high sweat chlorides. And we want to know why. We want to know where that's coming from. Additionally, we harvest, we get nasal brushings. It's a fairly invasive process. If we can do the same testing using a skin biopsy, maybe there's another option. That's the thought there.
Hi, Lane Clark, Missouri. You put on the carbachol first and then use the milleride to inhibit it. Did you ever not put the milleride on first, or put it on first and then see if you had
Right.
calcium activated chloride channels?
Right. So, I'm sorry. That last part again, the calcium activated.
Well, if you put on the milleride first to block ENaC, and then put on your carbachol to see if you had any residual response there.
Oh, interesting. Right.
It might show some of that response is calcium activated chloride channel?
Yeah, so we've done a lot, we've done a lot of optimization. So one thing we've done is we've tried to use DIDs for the calcium activated chloride channels. We didn't see a whole lot. There are supposed to be a significant number in the coil of those calcium activated channels. Yeah, so for H&Es, you don't use the carbachol because you have a high, level of ENaC just normally, and we do need to stimulate it for the sweat glands. It really does tend to come right back to baseline. There's not a whole lot of missing current to account for, but that's a really good point. So you're thinking just swap the two, swap the order?
Just to see.
Yeah.
And there's also a specific inhibitor for TME 16A. So,
Yes.
So it's another option.
Yeah.
Thank you.
Thank you.
We do have a question in the app asking what cell types express CFTR in the cultures.
In the culture specifically. So that's a complicated question. Yeah. So CFTR is supposed to be, primarily expressed in the duct, and that's why we see the elevated sweat chloride in people with CF. In our staining, we're having a really hard time finding it in the duct. And here, so this is coil, where we see significant CFTR, and we see it at the apical face, and some of the cells express significantly more. I'm not finding anything in the coil, but there are a whole other host of issues that come about with the staining of CFTR, as a lot of you know. Yeah, so it's both coil and duct. The extent to which each is contributing is still a question. So we really do want to have smaller portions of the full gland in the culture. So hopefully the duct cultures will help answer that question.
Hi, Bruce Schultz from Kansas State. Really nice talk and great system here. A lot of, first of all, what was your baseline transepithelial resistance on the cell, these types of cells?
You guys are letting me use all my extra slides. Yeah, so the resistance increased over the course of a month. The ones we were really happy about, we were looking at 2,000 micromoles.
Good things come to those who wait.
Exactly. And these guys are significantly slower than H&Es to grow and divide onto. It takes more time.
So a lot of times, transepithelial resistance is enhanced with using corticosteroids like Dex or cortisol or something like that in your medium. The thing that goes with that is quite often you also get an upregulation of ENaC expression and activity. Are you using corticosteroids at all? And if you do or don't, can you modulate ENaC activity in this system?
Interesting. So again, I don't want to go too much into the media composition, but we do use hydrocortisone, which is a corticosteroid, right? Yes. And so during optimization, we did use slightly different levels, but I don't think we actually checked ENaC. Well, we've only recently got to the point where we can actually put these filters onto the osing chambers, so I don't think we've checked that. No, but that's a very interesting thought. Thank you.
We have one question over here, and then we'll hit the microphone.
Right. Right, and I'm sure you'll hear also some issues with CFTR detection and single cell in the next talk, because CFTR is particularly prone to drop out due to its low expression levels. So you're really only seeing it in the higher expressing cells. I was seeing CFTR in highly expressed in some subset of ductal cells. But I'm really expecting there to be a good amount of dropout there, because there is known to be lower CFTR amounts in the coil. So the dropout is going to be more frequent in the coil. So that comparison probably can't be made by that. But if one did bulk and separated the two parts, so bulk seq with duct and with coil, you could probably get a better answer to that. though I don't want to extend my PhD to do that right now.
Okay, Yunjie from Indiana. Very nice talk. So my question is, have you done any correlation about the CFTR function detected in these sweat cells and the patient's phenotype?
Oh, yeah. Yes. So correlation between sweats and activity is what you're asking? Yeah. Yes, we see a good correlation. And I was showing that with just the individuals with F508del. But yes, we do see a high correlation. Yeah. Thank you. Thank you.
Fantastic. Up next, we have Kevin Zhu, a second-year medical student from the University of Virginia who's interested in using next-gen sequencing methods to identify treatment targets. We'll be speaking on single-cell multiomics, revealing profiling reveals novel regulatory networks in the kidney. And let me get you set up. Okay.
All right. Good morning, everyone. I'd like to thank you guys for coming in today to this workshop. And I'd also like to thank the NACFC organizers for inviting us this year to present our data. My name is Kevin. I'm a second-year medical student from the University of Virginia, presenting this data on behalf of the Urban and Zhang labs at UVA. So today I'd like to present some findings from our ongoing project. Our goal is to use single-cell genomics to reveal the functional and molecular changes in the kidney within people with CF in a mouse model. Okay, I got nothing. So we know it's been a very exciting couple of years when it comes to the treatment and long-term management of cystic fibrosis. We now have these high-efficacy modulators like the CFTR drugs, and we have this huge and fantastic community of research that has thus so far helped millions of people with CF to live longer and fruitful lives. With these advances, though, we're also seeing people living long enough to see the extrapulmonary manifestations of CF further down the line. And one of those includes chronic kidney disease. So we're not entirely sure how this happens mechanistically, but we do know that the incidence of end-stage renal disease approximately doubles every 10 years of follow-up, and it contributes to about 13% of deaths within the cystic fibrosis community. So a bit about the kidney. The average person has just under 2 of them. And these guys filter a lot of blood, almost 300% of our total volume daily. And it's a major player in maintaining the balance of solutes in our plasma, so it's worth it doing a little bit of review. If I can get that laser pointer up. Yeah, so you start you have the nephron, which is the main workhorse of the kidney. This is where plasma is released into the nephron lumen at the glomerulus, filtered in the proximal tubules, the loop of Henle, up into the distal convoluted tubules, and finally excreted after the collecting duct as urine. Within those renal tubules, a lot of things happen. You get resorption, secretion of a lot of organic compounds. You get regulation of acid-base status. You get excretion of metabolic waste products. But most importantly, you have the maintenance of key electrolytes like chlorine and water. And where does CFTR play a role in all of this? Well, we see that in both humans and mouse models, CFTR is robustly expressed throughout the renal tubules. We have an example of one here. One area of the tubules I'd like to highlight is the proximal tubule. This is where CFTR is expressed very highly and plays a very important role in the endocytosis of proteins, which should not be in the urine. In functional studies, if we knock that out in mice, you observe this defective endosomal trafficking and you find low molecular weight proteins in the urine, which is phenotypically not unlike other genetic kidney diseases that lead to renal failure. CFTR is also found in the collecting ducts, particularly in beta intercalated cells, and it's believed they play a role in secretion of bicarbonate into the urine alongside this chlorine HCO3 exchanger called pendrin. Again, if you knock that out in mice, you see them develop a metabolic alkalosis partially due to that decreased means of secretion. So this is another example of this lung-kidney interplay when it comes to managing acid base, and a mutation like this potentially is worth investigating. So what does this all mean? The lungs and kidneys are fundamentally interconnected, and patients with CF, people with CF, are particularly susceptible to kidney injury in a number of ways. Whether that's the intrinsic consequences of losing CFTR in the kidney, or whether if it's the systemic consequences of cystic fibrosis, the malnutrition, the pulmonary dysfunction, all of which can lead to dysregulation of homeostasis in the kidney. So this is a very important research topic, and our goal in this project was to use high throughput sequencing at a single cell resolution in order to develop a single cell RNA pipeline within a CFTR knockout mouse model to study the molecular impact of CFTR knockout on the kidneys to better describe and characterize the risk of kidney disease in people with CF. So the mouse model that we used was developed by our collaborator, Dr. Craig Hodges. You might have heard of him before, and the rest of the Case Western Cystic Fibrosis Mouse Model Core. We used their G54X stop gain mouse model, first introduced in 2018. And we know this is a really robust mouse model for a number of reasons. One, it greatly decreases the level of CFTR mRNA in the lungs, the nasal epithelium, GI tract, the kidney, decreases CFTR protein activity. And it also leads to the classic signs of CF in the affected mice, including intestinal obstruction and a decrease in growth and survival. So our single cell experiment consisted of 2 mice, 2 from wild type, 2 from knockout, and 1 male and 1 female. At around 6 to 11 weeks, we harvested those kidneys and sequenced them using the Chromium single cell assay to generate single nucleotide RNA-seq data sets. We then aligned them to the mouse genome. We counted them, filtered them for quality, and then we used Seurat to cluster, annotate, and do differential gene expression analysis. So like my colleague Alice very eloquently gave an introduction to, this is another UMAP. This is a single cell UMAP, which is a 2D representation of our data set so far. After annotation, we identified 16 biological clusters of cells, 13 representing all of the major renal tubule segments, and 3 of them representing injured proximal tubule cells, proliferating cells, and immune cells. So naturally, when you get a data set like this, you want to know what's the expression levels of CFTR in the renal tubules. And exactly what was said before, we see quite low levels of CFTR counts at the transcriptomic level in the renal tubules. However, the counts that we do see are primarily concentrated in the proximal tubule segment. We also then looked at any global changes in gene expression in each tubular segment. We see that not only are there changes to the proximal tubule, but also other tubules down the line, the ascending loop of Henle, the distal convoluted tubules, et cetera, et cetera. This could be a result of compensatory changes as a result of CFTR ablation in the kidneys, or we could see these as a result of the more systemic manifestations. So to sharpen the focus down for the purpose of this talk, we turned to several gene sets described in this 2022 paper, which outlines several key transporters and regulators of the major renal functions shown above. And here is a heat map, kind of representative heat map of the expression changes for those genes I showed previously. And starting off, we have the gene set regarding sodium, chloride, and potassium transport. And the asterisks are highlighted as a representation of significance, significantly changed genes. And we see quite a few significantly changed genes here, primarily in the proximal and distal tubules. Here's a summary of what we see. We see an increase of sodium, potassium, and 2 chlorine channels in both the proximal and distal segments. We see a slight decrease in the sodium chloride transporter in the distal convoluted tubules. We see a decrease in carbonic anhydrase and the NAH exchanger, increases in the angiotensin 1 receptors, increases in potassium channels involved in potassium resorption. And we also don't see changes in ENaC, which is typically regulated by aldosterone. But what does this all mean? Because there's a lot here. This all points towards the inward movement of sodium and thereby water, as well as potassium back into the blood. And all of those things we know are regulated or are involved in blood pressure regulations. This doesn't necessarily mean that there's an increase of risk of hypertension in people with CF. In fact, they typically have lower blood pressures phenotypically than their peers, likely through that decreased water and salt content through sweat. So this is an expected compensatory mechanism seen in the kidneys. On the topic of fluid balance, we also looked at the genes related to acid-base and water transporters, and we see that in the proximal and the distal convoluted tubules specifically, we see an increase of aquaporins as well as an NAHCO3 cotransporter. So what does that mean? We see the increases of two transporters fundamentally needed to resorb water back into the bloodstream, just like we saw in the previous gene set. So increased bicarbonate resorption, decrease hydrogen ion excretion, and increased water reabsorption. In this gene set of other important ions, we see, interestingly, a great decrease, a very consistent decrease in the vitamin D receptor in the proximal tubules. But we also see an increase in calcium-binding proteins. There's a lot of potential causes behind this, but I'll just leave it at this. There's a lot of changes to the regulation of calcium, phosphate, magnesium, and vitamin D response. And then finally, when we turn to organic compounds and toxins, we see consistent decreases in a number of transporters in the proximal tubule. We see decreases in the urate transporter, URAT1. We see decreases in two critical genes like the multi-drug resistant associated proteins 2 and 4. And these are decreases in those transporters may decrease the excretion of organic ions, such as many different drugs, chemotherapy drugs, antibiotics, statins. But what's interesting is that those decreases we see in the proximal tubules, we see compensatory increases in the distal convoluted tubule. So we see a disruption of a lot of transporters regarding drug excretion, organic ions, excretions that opens up a lot of potential avenues into the pharmacokinetics of drugs within people with CF. So just a summary, the data set here is very rich, but the data we've shown so far demonstrates quite a few things. We see an increased renal salt and water reabsorption. We see an increase of bicarb absorption, as well as a decrease in hydrogen ion secretion. We see this overall just dysregulation of calcium and vitamin D response. And we see a disruption of organic compound secretions and absorptions, which may alter the pharmacokinetics and predispose individuals to other metabolic syndromes. And it's important to note that it's hard to say right now whether the things that we're seeing in this data set are directly caused by CFTR knockout within the kidney, or whether they are the consequence of systemic effects of CFTR dysfunction in the animal model that's being combined with CFTR loss in the kidney. Nevertheless, this data here is again very rich and offers a mechanistic insights into what we see in the animal models, and more importantly, what we see clinically. And we plan to dig very deep into these mechanisms with more samples and more experiments in the future. I'd like to thank everybody involved in the project, including Matus and Dr. Sean Hu for their support and mentorship, Dr. Urban and Dr. Zhang for their this opportunity to work on this project, and our collaborators, Dr. Hodges in the Case Western CF Mouse Core, Dr. Gardner from Dartmouth, and Dr. Harris from UAB. And yeah, thank you for coming, and happy to take any questions now.
Great talk, Kevin. So there is a known interaction between ADPKD and loss of CFTR, where it's actually loss of CFTR is protected. Does your data set kind of get to why that might be?
It's hard to say whether there is a direct causative link between a loss of CFTR and the decreased risk of APKD. I can say though that the mouse model specifically, what we were sequencing were the survivors of that G542X, that animal model. So that's something we're planning to look into, but that's a very valid point.
We have a couple of questions in the app. First, have you detected any changes in glucose transporters in your model?
Sorry, can you say that again?
Any changes in glucose transporters?
I can't say from the top of my head. I do recall one of them appearing in the proximal tubule, but the exact one I cannot comment on right now.
And the second question, why single nucleus and not single cell?
That's a good question. Definitely something I'm going to look more into. We have a poster session, I think, on Saturday, and by then I'll probably take a lot of these questions, do a little bit of reading on my own. I think that was just the single cell kit that we went with. I think it was the Chromium multi-omics model, multi-omics single cell kit. So I think by default they use single nucleotide.
Excellent talk. There are two splice variances of SLC4A4, one of which is definitely predominant in the kidney. Did you look for the other variant at the same time?
We did not, but that point taken, definitely something we should take a look into. Mm-hmm.
Then the other question is, MERP4 is also a prostaglandin transporter. It's required for prostaglandin exit from cells. And I noticed it was on your list as well. Have you looked at COX-2 or any of the prostaglandin pathways to see if they're modified?
No, we did not either. These are great suggestions, by the way. Yeah, definitely we'll take those into account.
I have another question for you. Any thought on using any nephrotoxic meds in these mice and then repeating this kind of analysis?
This is a really great question for multiple reasons. Because one of the things we considered was that one of the risk factors of kidney disease within people with CF is the repeated use of aminoglycosides, which are very well known to be nephrotoxic. It's definitely something that we're thinking about, but we're also thinking about it for another reason. One of them, that specific G542X mutation can be actually read through completely by a specific aminoglycoside, I think gentamicin. So what we might end up seeing is not only the risk of nephrotoxicity, but also protective, like a CFTR read-through, bring back of function with the use of aminoglycosides. So it's definitely something very interesting that we want to look into.
Well, thank you. So our final speaker of the session is going to be Sarah Mansbach, who's coming from Case Western Reserve University as part of the scientific staff there in the Department of Genetics and Genome Sciences. She'll be telling us how myokine production in CF mouse skeletal muscle differs from the wild-type controls.
Good morning, everyone, and thank you for coming so early, first day. I would also like to thank Dr. Cook and Dr. Kramer for choosing my talk to be presented to everyone. So my name is Sarah Mansbach, and I'm an RA3 at Case Western Reserve University, and today I will be discussing the differences in myokine expression observed in the skeletal muscle of wild-type and CF mice. Oops. I have no relationships related to the presentation to disclose. So a strong correlation has been established between lean body mass and lung function. The correlation that has been established is a positive one, in which an increase in lean body mass is associated with an increase in lung function. Now, prior to the implementation of modulators, people with CF had difficulty in putting on and maintaining lean muscle mass. However, since modulators have become a form of therapy, people with CF have been experiencing increases in lean body mass. However, despite the increases that are being observed, the body composition of people with CF still does not reflect that of people without CF. Now, in our multiple studies in which we have utilized the CF mouse model, we have found that the body composition of the CF mice is similar to that of people with CF. Both people with CF and the CF mice experience a reduced lean muscle mass in addition to a reduced peripheral fat. And one thing that we observe in the CF mice is that even when you put them on a high-fat diet, they do not experience any changes in body composition, more specifically to those related to subcutaneous and visceral fat. Now, with these differences in body composition in mind, we wanted to look at skeletal muscle to see if something was happening in the skeletal muscle that would attribute to the phenotypic differences that we see in our CF and wild-type mice. Now, we decided to look at skeletal muscle because it is a metabolically active tissue. It produces myokines that have both autocrine and paracrine effects. And for this study, the myokines that we chose to look at are ones that are involved in muscle growth, metabolism, and inflammation. To begin the study, we started by looking at RNA-seq analysis performed by our colleague, Dr. Aura Perez. And in her analysis, Dr. Perez used the gastrocnemius muscle of 6 male wild type and 6 male G542X, aged about 7 weeks old. In her analysis, Dr. Perez found that despite the absence of CFTR in this muscle, that of the 24,000 transcripts that are present, approximately 7,000 of them differ significantly in expression between wild type and CF mice. So that's about a 30% difference in transcript expression. So of the 7,000 transcripts that differ, approximately 3,600 of them are upregulated in the CF mice, while about 3,500 are downregulated in the CF mice. So with this information in hand, we went on to choosing our mouse models. So of course, for our control, we use the wild-type mice. These mice do not manifest any CF phenotype. We also chose, for our CF genotypes, we chose the R117H mouse model. This mouse produces a more mild CF phenotype. And then we also utilized the Delta F and the G542X mouse models, both of which present with a more severe CF manifestation. As you can see, if you look at figure 4, the yellow line is representative of the R117H mice, and you can see that these mice have a better survival rate than the Delta F or the G542X mice. And if you look at figure 5, which shows intestinal oosing data that looks at the short circuit current, when you look at the function of CFTR, although the R117H mouse's CFTR function does not match that of the wild-type mice, they do have more CFTR function than both the Delta F and the G542X mice. So with our mouse models in hand, we wanted to profile myokine expression and histological differences that are present in the skeletal muscle. So we took our wild-type Delta F, G542X, and R117H mice, and we harvested the quadriceps from these animals. And utilizing the quadriceps, we either isolated the RNA and performed qPCR to look at a wide variety of myokines, Or we took the quadriceps, fixed it, and performed immunohistochemistry to look at the distribution of type 1 and type 2 muscle fibers. We also wanted to look at the impact of infection on myokine expression. So to perform our infection studies, we took wild-type delta F and G542X mice, and we administered either sterile beads or beads that were laden with PAM 5715, which is a clinical isolation of Pseudomonas aeruginosa. We administered these beads to the mice via bronchoscopy. 3 days after bead administration, the quadriceps was harvested from these animals. We chose the 3-day time period because if there's going to be an inflammatory response, the peak of it would occur at 3 days after bead administration. So with the quadriceps in hand, we once again isolated RNA and we performed qPCR. And for the purposes of this discussion today, in regards to the infection studies, I will be focusing specifically on IL-1 beta and IL-6. Now, as I said, we looked at a wide variety of myokines. However, for today's talk, I will be focusing on the myokines with a little yellow star by them. We have begun studies on the myokines ATF3, BDNF, and IGF-1. However, I will not be discussing them today, seeing as though our studies with those myokines are ongoing. Now, the first two myokines that we looked at are ones that are involved in the regulation of muscle growth. Myostatin is a myokine that is involved in the inhibition of muscle growth. Now, we hypothesize that since CF mice are smaller than wild-type mice, that myostatin would be upregulated in these mice. However, if you look at the figure on the left, which shows the full difference in myostatin expression between wild-type delta F, G542X, and R117H mice, we found that there is no significant difference in expression of myostatin amongst any of the CF mice and then the wild-type mice. However, when we looked at IL-15, which is involved in promoting myoblast differentiation, we found that when you compare the wild-type and the R117H mice, there's no difference in IL-15 expression. However, the delta F and the G542X mice have a greater expression of IL-15 than the wild-type mice. Now, with this upregulation on IL-15 in the delta F and G542X mice, we should be seeing an increase in muscle growth. However, we are not, and we are currently hypothesizing that that is due to the inability of the skeletal muscle to simply energetically accommodate that growth, because mitochondrial function in CF has been shown to be dysregulated. We are also hypothesizing that there might be competing signals with IL-15 that prevent the muscle growth that IL-15 would try to make happen. So next we looked at myonectin, and myonectin is involved in the regulation of lipid metabolism, and it does so by acting as a signaling molecule to adipose tissue to release fat. Now, we looked at myonectin because lipid metabolism is known to be dysregulated in CF. And in studies that have utilized a myonectin knockout mouse, they have found that when these mice are fed a high-fat diet, they accumulate fat in adipose tissue as one would expect. However, in our CF mice, what we find is that when you put them on a high-fat diet, they do not accumulate fat in the adipose tissue as one would expect. As you can see in figure 8, if you look at the two far right mice that were fed a high-fat diet, the wild-type mouse does accumulate fat in the adipose tissue, where the CF mouse really does not. Instead, what we find is when the CF mice are put on a high-fat diet, they accumulate fat in the liver, which you can see in the bottom panel of figure 9 by the fatty liver. So in looking at myonectin expression, what we found is that once again, the R117H mice compared to the wild-type mice do not exhibit any increased or decreased expression of myonectin. However, the delta F and the G542X mice do express myonectin at a higher level than the wild-type mice. And we are hypothesizing that this increased expression of myonectin could contribute to why the CF mice are unable to accumulate subcutaneous and visceral fat, and perhaps why instead we observe the fatty liver. Next, we looked at IL-6 expression, and IL-6 is a myokine that is known for promoting inflammation and also muscle atrophy. So if you look at figure 10, we found that without infection, IL-6 is upregulated in the delta F and the G542X mice. However, when you add infection in, which is represented by figure 11, we find that when you compare the infected wild-type mice to their non-infected counterpart, there is a significant upregulation in IL-6 expression. However, when you look at the infected CF mice compared to their non-infected CF counterparts, there is no significant upregulation or downregulation in IL-6 expression. So this suggests that basally IL-6 is already being upregulated in delta F and the G542X mice, and that it does not respond to infection. Next, we looked at IL-1 beta expression, and IL-1 beta is another myokine that regulates inflammation. And looking at figure 12, we can see that basally IL-1 beta is not upregulated or downregulated in any of the CF genotypes when comparing them to the wild-type mice. However, we find that when infection is introduced into the story, that IL-1 beta becomes quite significantly upregulated in the CF mice. It's quite a drastic upregulation that we observe. So this suggests that with infection, IL-1 expression becomes hyper-responsive to infection in our CF mice. So in addition to profiling myokine expression, we also wanted to look at the histological differences in the skeletal muscle of our wild type and our CF mice. So we performed immunohistochemistry to look at type 1 and type 2 fiber distribution, and our initial studies have found that in the CF skeletal muscle, there is less of the type 1 fiber. However, we need to perform more studies to really draw a firm conclusion on this. So in conclusion, we have found that there are significant myokine expression differences between wild type and CF mouse skeletal muscle, despite the absence of CFTR expression in the skeletal muscle. And while myokine levels are not different in the R117H mice compared to the wild type mice, this does suggest a role for CFTR function in regulating skeletal muscle myokine expression through either a direct or indirect mechanism that has yet to be established. There are a lot of directions in which we would like to take this study. To begin, we would like to perform more immunohistochemistry to elucidate further histological differences in the skeletal muscle of our wild type and CF mice. We would also like to utilize a myonectin knockout mouse model and cross that with a CF mouse model to see if knocking out myonectin in our CF mice corrects any of the lipid metabolism dysregulation that we observe. We also intend to perform modulator studies and exercise studies to see if either of these factors affect myokine expression in these mice. And using a biobank of serum from our colleague Dr. Kim McBennett, we would like to perform human studies to look at myokine expression, especially in patients pre, during, and post-Trikafta. And furthermore, we would like to elaborate on our infection studies to see what other impact infection might have on myokine expression. I would like to thank all of my colleagues at Case Western Reserve University, for without their help and their support and their input, this study would not have been possible. And I would also like to thank the Cystic Fibrosis Foundation for their generous financial support. And again, thank you all for coming, and I'm happy to take any questions.
So we have a couple questions in the chat that we'll start with. The first one is IL-6 is a pro-inflammatory cytokine. However, when stimulated by the muscle, it actually increases metabolism and reduces TNF-alpha, hence inducing an anti-inflammatory response. Have you considered looking at how IL-6 following muscle contraction differs between those genotypes that you studied?
I don't think that is something that we have quite considered yet, but that is certainly something to take into consideration, especially in performing exercise studies. I think that would really elucidate that effect.
And then following up with that, myokines are produced from skeletal muscle. Without stimulation of the muscle, how can you be sure that the production of the cytokines you looked at are actually being produced from skeletal muscle and not just deposited in the muscle that yields the expression?
That is a very good question. Once again, I think that is something that exercise studies would further elucidate on. I will say with these mice, when the muscle was harvested, they had already been running around in their cages. So presumably there already was some muscle contraction going on. They weren't just sleeping or hanging out.
Following up on those, one of the, I think, more powerful aspects of your study that is truly impressive is the different genotypes that you encompassed in this, and the differences that you saw whereby the R117H genotype was more similar to wild type. Can you speculate then on whether or not you think that this is going to be a cell intrinsic effect versus other inflammatory markers that are feeding back on these cells?
That's a good question. I do think there perhaps could be a more cell intrinsic effect, but I think we're speculating in the lab that it's definitely more genotype dependent.
Another question. I saw a decent amount of spread, say, within the F508del group when looking at these myokines. And I'm just curious, did you ever look and see if that linked to different body weights within groups, or if the metabolism defects and the low body weights that these mice have are at all correlated to the myokine expression you saw? If that makes sense.
Sorry, if you could rephrase.
Oh yeah.
that.
So is the body weight of the mouse at all associated with the changes in the myokine production that you're seeing within a group?
Yeah. I will admit that we did not weigh these mice prior to harvesting muscle. I think that certainly would be a good aspect to look at in the future.
Yeah, I was also considering whether a gut-corrected model, that lacks some of the growth abnormalities, might help you distinguish what's driving the myokine abnormalities, given that there's no CFTR function that you're finding within the muscle itself.
Mm-hmm.
All right. Oh, please, go ahead.
Yeah, I was wondering about the gut-corrected experiment. Also to control for the systemic inflammation that these mice had, just because the gut. I assume all your mice are on laxative.
I'm pretty certain the delta F. Oh, she wanted to know if all of these mice are on a laxative, the CF genotype mice. To my knowledge, the delta F and the G542X mice are on a laxative because they are more prone to intestinal obstructions. The R117H mice are not, because they don't experience at least as high of an incidence of the intestinal obstructions as the delta F and the G542X mice.
When they can take it away.
Oh, if we take away, that would be a interesting thing to look at. However, we would probably have to get that approved through IACUC before we did that.
Just one more question to follow up. When thinking about specifically the stress that these cells are placed in, if it is a cell intrinsic effect that I would argue that maybe some of your data points towards, do you think that ER stress could be contributing to some of these inflammatory myokines that are produced? And how much do you think the cell is reacting to the environment, the inflammatory environment, whether it's the intestine or not, versus what's being produced by the cell? And can you measure these in culture?
Okay. Well, I know we have discussed possibly isolating these muscle cells and growing them in culture, which I think would certainly negate any of the effects that a mouse in a cage might experience. Our mice are very well cared for in our mouse facility, so I'm not exactly sure what stresses they might encounter. but it certainly could be affecting it. And I think we do need to look further into maybe any stresses they might be encountering.
All right, thank you. And thanks to everybody who came for this session. I think it was wonderful, productive, and hopefully you can enjoy the rest of the conference.